Hey, what's up, classmates? Today we're going to be doing cholesterol metabolism 2. This is very long. I'm going to try to keep this video under 30 minutes. Let's see if I can do it. All right, objective set A, or objective A, know the absorption of lipids, fatty acids, and cholesterol, and the subsequent metabolism of these lipids in the blood as a result of being transported by chylomicrons. Okay, before we do that, let's write some definitions. So a lipoprotein is going to be a complex of cholesterol, phospholipids, cholesterol esters, triacylglycerides, tags, and specific carrier proteins, so apolipoproteins. This is important in targeting specific tissues and targeting where a certain fat will go. Okay, so the content that make up a lipoprotein is what gives it its classification. So for example, chylomicrons are the largest of all of the lipoproteins, but they're also the least dense. And you have others that are named by their densities, like very low density lipoprotein, VLDL. So apolipoprotein is specific to the type of lipoprotein it's associated with. It's based on what roles it plays as targeting specific tissues. So here are two pictures of two lipoproteins. I don't know what this is. So the whole thing is a lipoprotein, and you can have these little apolipoproteins on the surface of it, and then inside you have your tags and your cholesterol and your cholesterol esters. This membrane right here, it forms a micelle, and it's made out of phospholipids. One last thing, some cholesterols actually turn into this cholesterol ester via this enzyme acyl-CoA cholesterol acyl transferase. We're just going to call it ACAT. And basically, it's used, cholesterol ester is used as a form to store steroids or like a cholesterol precursor, and also it's going to be transported in this form if its final destination is a place where it will be used for storage, basically. Okay, so let's take a look at how fatty acids and cholesterols are actually circulating in our body when we ingest it from our diet. So I actually drew this because it took such a long time. But I drew this because I couldn't find a good picture on the internet, and I really like this picture in our textbook. Sorry for the background noise. My family is eating dinner, and they're very loud. So I'm going to be navigating between this page. You can refer to your textbook if you would like, if that's easier. This is exactly like the picture in our textbook. Okay, so the first step. So we ingest fatty acids, right? And it's going to have a combination of fatty acids, we ingest lipids, and it's going to be a combination of fatty acids and cholesterol. So a fatty acid that is ingested will have a different fate than cholesterol. So your fatty acids are going to be broken down and then reformed into your triacylglycerols, tags, and they're going to basically form this chylomicron up here, this chylomicron down here, I guess you could say. And you're also going to have your three apolipoproteins, so ApoB48, ApoC2, and ApoE. And additionally, you're going to have some cholesterol in there as well that you ingest from your diet. Once this is formed, you're going to actually enter it into your lymphatic system, where it can target certain parts of your body, so like mammary glands or your muscle or your adipose. And what's going to happen is your ApoC2 is going to activate lipoprotein lipase, and you're going to release free fatty acids to these tissues. So what you're left with, right, is the remnants of what used to be a chylomicron. And so your chylomicron is going to be or your chylomicron remnant is going to consist of cholesterol, right? Because we released free fatty acids, but we didn't release cholesterol yet. And then we also used our ApoC2 to activate our lipase. So we still have our other two lipoproteins, B48 and E. And what it's going to look like is this, I drew it right here, remnant, and it has cholesterol in it. And it basically looks like this, but deflated, right? Because we sort of released all of these free fatty acids. So what's going to happen next is the remnants are actually going to travel to your liver through your lymphatic system, and then receptors on your liver are going to be activated by your apolipoprotein E, and it's going to take in your remnant chylomicron via endocytosis. From there, your endosome is going to combine with a lysosome and degrade, and you're going to degrade various enzymes and phospholipids. But the most important thing is you're going to actually release all that cholesterol that was left in that sort of deflated sac, and you're going to release all of that into the cytosol. Okay, so what's blue? That is the exogenous pathway. What we're doing next is red, endogenous pathway. The way I like to think about the exogenous pathway is first getting fatty acids to where it's needed most through your diet, and then from there getting cholesterol to your liver. So cholesterol can't actually be distributed right away. It has to go to your liver, and from there your liver is like a post office, I guess, and it's going to distribute the cholesterol where it needs to go. Your liver can also distribute free fatty acid to other places as well, so it's not just cholesterol. So yeah, let's talk about step 5, and this is the first step of endogenous pathway. So now your liver has a bunch of fatty acid and cholesterol, and let's say, for example, it has an abundance of it. So to mobilize your fatty acid that's in your liver, we're going to have to make them into tags, tags. And then the cholesterol, some of it's going to be turned into cholesterol esters with our ACAT enzyme, right? And then from there, we're going to form a new lipoprotein. It's going to be called VLDL, very low density lipoprotein. And this is going to be sent to different parts of your body. It's going to enter your lymphatic system first. So this is not as low density as chylomicrons, but it's still very low density. Okay, so the apolipoproteins associated with the VLDL is going to include ApoB100, ApoC1, ApoC2, ApoC3, and ApoE. So right now we have something very similar to a chylomicron, except it's a little bit different, right? It's a different type of lipoprotein, but its contents are essentially very similar. Okay. So step 5, that's this right here. We're sending out our VLDL into our lymphatic system. All right, so step 6. Essentially what's going to happen is you're going to keep releasing free fatty acids, right, until all you're left with is cholesterol and cholesterol esters. So step 6 is where when tissue that need more tags, right, more triacylglycerides, you're going to send your VLDLs to those tissues, and we have ApoC2 acting once again, because similar to the chylomicrons, and the ApoC2 will activate lipoprotein lipases and release free fatty acids. When you do this, you're going to deflate it a little bit, and what you're left with is it's called an intermediate density lipoprotein. All right, and you're basically just going to do it more and more and more, and your remnant is going to be mostly cholesterol and cholesterol ester, and now this remnant is known as LDL. So that's shown right here. You're in your VLDL is going to your lymphatic system, and it's going to release FA, free fatty acids, and continue to do that until you reach your LDL. And now your LDL is just basically cholesterol and cholesterol esters. Okay, so now we have an LDL as opposed to VLDL, so not a very low but a low density lipoprotein, and it's going to carry cholesterol to tissues that need it. So like muscle or adrenal glands or adipose tissue. And the way it does this is so your cells that need this cholesterol, they're going to form a bunch of receptors, and these specific LDL receptors are activated by ApoB100. So remember, our free fatty acid tags were activated by ApoC2. This one's going to be activated by ApoB100. So step 7, let's look at that. Yeah, right here, right? So it's going to go to your like adrenal glands or muscle or adipose, and the way it gets activated and released into these cells is your ApoB100. All right, 2 more steps in the endogenous pathway. So not all of your LDL is going to be used up, right? You're still going to have a lot of cholesterol in that remnant, your VLDL remnant, right? So actually some of it is delivered to macrophages, and this is going to convert your macrophages into like foam cells, which are just macrophages with a bunch of cholesterol in it. And then this later we're going to learn it's going to enter the reverse cholesterol transport, which is purple in this diagram. We'll talk about that later though. And then the last step, not all of it is turned into foam cells, right? Some of it is actually still going to be in your LDL, some cholesterol, and it's going to return back to your liver. And now LDL receptors on your liver are going to take in the LDL, your low density lipoprotein, right? And now the remaining cholesterol can be used for stuff like membranes or bile, or you can re-esterify it for storage using our ACAT, remember? So yeah, before we move on. So this, once again, just recap, I like to think of this as the post office. We need to get cholesterol to the liver so that the liver can distribute it. Whereas for fat, when we eat it, it can go straight from our digestive tract into like muscles or mammary glands or adipose. So essentially, cholesterol takes a more roundabout way of doing things, but fat can also take a more roundabout way of doing things, but they have the option of taking this shortcut right here. Okay, so objective B, the 3 major lipoproteins present in flaccid blood and metabolism of lipids associated with these lipoproteins. We saw this earlier with this diagram, right? So it's going to be these lipoproteins associated with our chylomicrons right here. So ApoB48, ApoC2, and ApoE. So ApoC2, if you remember, is going to activate our lipoprotein lipase and release ingested fat as free fatty acid to desired tissue. This can be used as storage, or it can be used as energy, like in muscle. And then, so now we have remaining remnants that contain ApoB48 and ApoE, and these two are going to play a role in providing clearance for transport to the liver. So the way I think it works essentially is, if you have a high density of ApoB48 and ApoE on your chylomicron, and a lot of your ApoC2 has been used up, then that's a signal for it to be ready and cleared for transport to the liver, where cholesterol can be distributed. Additionally, ApoE also works as the lipoprotein that helps the chylomicron get into the cell via endocytosis. Objective C, the flow of cholesterol and fatty acids through body using forward cholesterol transport and reverse cholesterol transport. So in objective A, we actually already did this forward cholesterol transport, and that was essentially the exogenous and the endogenous pathways. So now we're going to talk about the reverse cholesterol transport, which is purple in this diagram, and it is called the reverse cholesterol transport. So essentially what's going to happen is your liver is going to produce this HDL, your high density lipoprotein, and it's going to be in a nascent form, so it's not fully matured yet. And this nascent form does not yet contain like any cholesterol ester, or like it has very scant amounts of cholesterol itself. The major apolipoprotein is ApoA1. Sorry if that's hard to read. This is A1, like this steak sauce. And also it's going to contain an enzyme LCAT. So we talked about ACAT earlier. This one's LCAT. It's called lecithin cholesterol acyltransferase. Lecithin is a fancy name for phosphatidylcholine. So I don't know if you guys remember what phosphatidylcholine looks like. You really should though, because it was in complex lipids. And essentially this LCAT, lecithin, I don't know if I'm saying it right, cholesterol acyltransferase is going to turn cholesterol and phosphatidylcholine into a cholesterol ester. So essentially your HDL is going to use LCAT to produce cholesterol esters, right? We said that just right now. But it's going to use precursors from your very low density lipoproteins and chylomicron remnants. So after your VLDL uses this has done its job, and it's like and your chylomicron has done its job, is going to leave behind a remnant, right? And this remnant is going to have some cholesterol still in it, and it's going to have its phospholipids and its phosphatidylcholines in its membrane. And essentially this LCAT is going to turn all of these into cholesterol esters. So a good way I like to think about this step is your forward step is like a sloppy way of getting fatty acids and cholesterol to places in your body. It does it quickly, but it's fast. And then now the reverse process is cleaning up after it and reusing some of the things that it has left behind, if that makes sense. So in this step, this is what it's showing. You're forming your HDL, all right? Okay, so in addition to forming its own cholesterol esters from your remnants of chylomicrons and VLDLs, your nascent HDL can also accumulate cholesterol from extrahepatic cells, like foam cells, for example. So that's this step right here. It's taking some cholesterol. Remember we said how this like accumulates cholesterol. This is actually taking some of it for itself. And your nascent HDL is going to form its own cholesterol esters and cholesterol itself, and is going to grow into a mature HDL. All right. So once it's mature, now it's ready to continue, basically. So now it's mature. It's going to release some of its cholesterol ester and transfer it to LDLs sometime. So that's this step right here, step 13. And then the rest of the time is going to come back to the liver through your SRBI receptors, so scavenger receptors. So remember how I said this reverse process is kind of like cleaning up. So a good way to remember that is this SR stands for scavenger receptor. So, and then this last little step right here, the intrahepatic pathway. Most of the cholesterol that returns from your reverse system is going to be used to create bile salts. That's basically all this is saying. This is reported here. So your reverse cholesterol transport is going to bring in via SRBI cholesterol esters, and it's going to be used and enter this bile salt pathway right here. All right, so objective D, know the metabolism LDL derived lipids after endocytosis. So Tara asked for 3 onwards. We're just going to go 1, 2, and then 3 onwards because it's not that much more. So in this first step, you're going to make these LDL receptors that are going to go to your cellular membrane. They're made by your rough endoplasmic reticulum, and then they're processed in your Golgi complex. And these receptors are going to be what is responsible for accepting your LDL particles and taking them via endocytosis. Okay, so then we said this earlier, right? Our ApoB100 will bind to these LDL receptors. and then your endocytosis occurs. And then step 4, LDL receptors are recycled. So once this guy enters, your receptors are they're gonna part off from your LDL, and they're gonna surface back up to the cellular membrane and be reused. Part 5, LDL inside your endosome fuses with a lysosome, right? We sort of talked about this earlier. And then the last step, lytic enzymes degrade ApoB100 and cholesterol esters to release cholesterol, amino acids, and free fatty acids to the cytosol. Okay, objective E, know how cholesterol regulates feedback inhibition of the steroid regulatory element binding protein SREB pathway. So here's the picture from our textbook. It illustrates it very well. Essentially, we're going to have 3 proteins that are bound together, our INSEG, our SCAP, and our SREB. And when they're bound together like this, forming the protein complex, they're going to be stuck to our endoplasmic reticulum. Essentially, what happens when cholesterol levels get, I think it's low. Yeah, when cholesterol levels get low, this NSEG is going to dissociate, and it's going to get transported down the ER, and then this little domain on your SREB is going to be an activator of a gene sequence that synthesizes various proteins needed for lipid synthesis as well as cholesterol synthesis. So the major regulator step for concentration of cholesterol is this step right here, beta-hydroxy beta-methylglutamyl-CoA, remember HMG, and it's going to turn to mevalonate. If you don't remember what this is, it's basically one of the initial steps in cholesterol synthesis, which is talked about in my last video, and the enzyme responsible for this is HMG-CoA reductase. So essentially, the concentration of this guy, or the inhibition of this guy, or activation of this guy, is going to largely determine the level of cholesterol circulating in our body. All right. So SREBs, steroid regulatory element binding proteins, work by activating target genes, including those responsible for HMG-CoA reductase. So remember earlier how I said this SREB, it has a domain that will dissociate and activate this gene sequence. One of those gene sequences happen to be the gene sequence responsible for production of HMG-CoA reductase. There are also other sequences. So these include receptors that are needed to uptake cholesterol and fatty acids. And then lastly, this NSAID has another function. It can sense when you have high levels of cholesterol. So right here, we talked about low levels of cholesterol. NSAID can sense high levels of cholesterol, and it basically ubiquinizes your HMG-CoA reductase. So yeah, draw this in your notes and read each step. I went through that really fast, but we did a lot of this in 445. It should not be very hard to understand, I don't think. Okay, so objective F, know the 2 regulatory mechanisms involved in maintaining whole body cholesterol homeostasis after an increased amount of dietary cholesterol is consumed. So when Dr. Bryan says after increased amount of dietary cholesterol consumed, this makes me think of short-term regulation. I don't know if this is the right answer because there were like 4, 5, or 6 regulation mechanisms proposed. But I think this is what he was aiming towards for this objective. So the major thing that, or the major way short-term regulation is achieved is through covalent modification. So phosphorylation of our HMG-CoA reductase. Remember, that's the key enzyme for regulating the levels of cholesterol in our body. Phosphorylation will actually deactivate it. So the first thing that phosphorylates it is low levels of ATP. So when you have low levels of ATP, you have high levels of AMP. And when you have high levels of AMP, you're going to activate your AMP kinase. And AMP kinase is going to phosphorylate our HMG-CoA reductase, which deactivates it. So when we don't have a lot of energy, when we don't have excess ATP, we're not going to be wanting to synthesize cholesterol, essentially. Same thing here. Glucagon stimulates phosphorylation of HMG-CoA reductase. When we have low blood sugars, it's sort of indicative of low energy, and this is once again going to phosphorylate our reductase, deactivating it. Opposite thing here, insulin. So we have high levels of blood sugar, we have a lot of readily available energy. This is going to promote dephosphorylation, which activates our HMG-CoA reductase. A little chart right here showing everything I just said. So insulin, glucagon, and ATP, these are going to be covalent modifications. Another regulatory mechanism responsible for saturated fat adversely affecting whole body cholesterol homeostasis. This objective I'm not very sure of at all. I think I'm going to wait until the lecture, and hopefully that will clear things up. But this is basically my hypothesis of what I think Dr. Bryan was trying to ask. So we mentioned covalent modification, right, with AMPK and insulin and glucagon. So I think he was sort of pointing towards this regulation right here, although it doesn't have adverse effects at all, because when levels are very high, it basically stops the production of reductase and it lowers our cholesterol level. So I'm not sure what he really meant by this objective. But the other major regulation that we haven't talked about yet was this liver X receptor, LXR. And it's a nuclear transcription factor, so an activator similar to SREB. My computer is lagging so hard right now. But the difference is SREB is activated when we have low concentrations of cholesterol, whereas this LXR, it's activated when we have high concentrations. So our LXR is going to be bound to our RXR, which is retinoid X receptor. And when cholesterol concentrations are low, it's not going to activate this transcription factor, and essentially no transcription happens. But when we have high levels of cholesterol, this is going to lead to high levels of oxysterol. So that's what you see in this right here, oxysterol. And this is going to lead to LXR becoming activated with our RXR, and this is going to activate transcription, and essentially the end result is proteolysis of HMG-CoA reductase. So when this is high, this stimulates the degradation of our HMG-CoA reductase.